The last section of notes for Chapter 12 is on the genetic code and translation. So let's talk about how does everything code first, and then we'll put it all together with translation. Remember that a codon designates an amino acid, and a codon is 3 nitrogen bases put together. So once again, a codon is 3 nitrogen bases put together. An amino acid may have more than one codon. There are 20 amino acids possible, but 64 different codons for those 20 amino acids. Some codons tell the ribosome to stop translating, and as we recall from the previous notes, AUG is the start codon. How do we figure out the codons? Well, we use the code by reading from the center to the outside. So when we're looking at a code, you start here, you move to here, and then finally you move to the third level. And then what you're going to end up with is your amino acid. So you start at the center and read towards the outside. So methionine is AUG, and so we go with AUG, and we end up with methionine. So let's go through and practice. What would GGG be? So we're going G, G, G. UCA, CAU, GCA, and AAA. You were able to determine that UCA was serine, CAU was histidine, GCA was alanine, and AAA was lysine. Remember the complementary bases. On DNA, A goes with T and C goes with G. On RNA, A goes with U and C goes with G. It's very important that you remember the base pairings when it comes to doing protein synthesis. Here we go. Protein synthesis, once again, is the making of proteins on the ribosomes. So the production or synthesis of polypeptide chains or proteins. There are two phases to protein synthesis. The first one is transcription, which we talked about in the previous video, and then there is translation, which is going to conclude this particular chapter. Once again, messenger RNA must be processed before it leaves the nucleus of eukaryotic cells. We talked about that in transcription. So how do we make a protein? We start with DNA, and if you remember, DNA is in the nucleus. Then we go to messenger RNA. Messenger RNA leaves the nucleus out the pores and goes to the cytoplasm, eventually reaching the ribosomes. Transfer RNA is located on the ribosomes, and finally, then we make a protein. So this on the ribosomes is where messenger RNA lands and pairs up with transfer RNA to make a protein. So here we go. Proteins are made of amino acids linked together by peptide bonds. And here's a whole nother bond, which we're reviewing again, peptide. That links together amino acids. We talked about hydrogen bonds linking together in the nitrogen bases back in DNA. So don't mix those two up. There are 20 different amino acids that exist. And amino acid chains are called polypeptides. Remember, poly means many. A segment of DNA that codes for the amino acid sequence in a protein is called a gene. There are two parts of protein synthesis: transcription, which we talked about in the previous podcast, that makes an RNA molecule complementary to a portion of DNA. In translation, this occurs when the sequence of bases of messenger RNA directs the sequence of amino acids in a polypeptide. So translation is making sense of what we transcribed or made a copy of from the DNA in the nucleus. DNA contains a triplet code. Every 3 bases on a DNA strand codes for 1 amino acid. Each 3-letter unit on messenger RNA is called a codon. It's very important that you get all of this vocabulary straight when we're going to make a protein. Most amino acids have more than 1 codon, which we saw in the wheel. Once again, there are 20 amino acids with a possible 64 different triplicates. And the code is nearly universal among all living organisms. This is a different way of representing the wheel. And if you look at this, you just have to figure out how to read it. The first base goes along this way. So if you're reading something, the second base is here, and then you've got the third base over here. So if we were coding for AUG, you would go A, and then you're looking in this column here, because this is the U. And then you go over to the column over here to find the G. So we put it all together, A, U, G. And we end up with our start codon for methionine. That's how that table is read. Transfer RNA, or tRNA, the amino attachment site is at one end and the anticodon is at the other end. So methionine is going to be the amino acid that's going to be attached to our transfer RNA. The anticodon, remember, is the opposite of the codon. So if you look down here, the anticodon, we know methionine is AUG. You'll notice that the anticodon, or opposite, is U, A, C, because A pairs with U, U pairs with A, and G and C pair together. So this would be the opposite. Okay? This AUG codes for this amino acid. Getting confused? Hopefully it'll all fall into place. So at the ribosome, we have what's called a P site and an A site, and this is usually what ribosomes look like. So what happens is in comes our strand of messenger RNA. And remember from the previous, AUG codes for methionine. So here's our first codon coming in. Translation is the synthesis of proteins in the cytoplasm. It involves the following: messenger RNA, which carries the codons, transfer RNA, which carries the anticodons, and the ribosomes. So those are the three pieces that are involved in translation. The amino acids are also present, and they are coming in attached to the transfer RNA. Okay? There are three steps to translation. First, we have to have a start codon, which is always AUG or methionine. Second, we add the amino acids onto our chain. And third, then we have a stop codon. You do not need to remember what the stop codons are, but I will expect you to remember that AUG is the start codon and the amino acid is methionine. So let's see how we're going to make a protein. The messenger RNA codons join the ribosomes. So in they come. And as you can see, the first one is AUG. So in comes our transfer RNA. Remember the bottom here, this is the anticodon. Up here is the amino acid. So transfer RNA comes in, as you saw it slide in. It brings the anticodon, which will match up, and we have our very first amino acid. There's your anticodon. They hydrogen bond. And in comes number 2. Now, as you can see, number 2 amino acid, your anticodon is going to be opposite your codon. So your codon is right here, your next 3. And you can see GAU is complementary to CUA. Notice there are no T's because there's no DNA. So in comes number 2. And what we have up at the top, if you look at the very top up here, there's 2 amino acids, amino acid 1 and amino acid 2. They're going to be joined together by a peptide bond. And there it is. So now in is going to come amino acid number 3. And because it is, your codon is CUU, you know your anticodon is going to be GAA. And there it comes. Notice that the old transfer RNA right here has left, and the new one is this is going to slide over, and then the new one is going to come in. And then if you look at the top, you can see that your peptide bonds are starting to attach all of your amino acids. Once again, the next codon or anticodon that's coming in is going to be GCU, GCU. Number 2 slides off. Number 3 is going to move over. And then we're going to have amino acid number 4. One thing that students always forget and get confused is the amino acid and the codon. These two match. The anticodon that's here does not code for the amino acid. So whenever you're coding for an amino acid, you use these letters here to determine which amino acid it is. In comes number 5. Number 4 is going to move over. So number 5 can then add. And this process continues to go on and on and on and on and on. And as you can see up here, we're at amino acid 200. In comes our stop codon, which says, all right, stop making this particular protein. We are finished. And you can see that there's going to be this long chain of amino acids, which ends up to be your protein. And that is the end. So once we have gone through all those steps, the end product is the protein. The end products of protein synthesis is a primary structure of protein, a sequence of amino acids bonded together by peptide bonds. And here's another example of how this works. Messenger RNA contains all of those codons, and it codes for this particular protein. Methionine is AUG, glycine is GGC, etc. And then we have our stop codon at the end. So the primary structure of the protein puts all of these amino acids together by peptide bonds.